I want you to imagine, just for a second, that you have a tiny, highly efficient executive assistant living right in the center of your brain.
Okay.
And it is this assistant's entire job to just look at the massive, overwhelming flood of memos coming in every single second from your senses, and then decide exactly what state your body should be in right now.
Right. Like, should you be running for your life or should you be fast asleep?
Exactly. Or, should you just be completely ignoring that distracting humming sound from the refrigerator in the background?
Yeah, which is an incredible amount of responsibility for one microscopic piece of neural real estate.
It really is. And welcome to today's deep dive, everyone. Our mission today is to explore a consensus review from a recent Brainstem Society meeting.
Yeah, and they've essentially mapped out the exact office where this executive assistant works.
Right. They are looking at a region of the brainstem literally the size of a pea called the Pedunculopontine Nucleus, or the PPN.
A PPN.
Okay, let's unpack this. Location-wise, where exactly is this executive assistant sitting?
Well, if we look deep inside the brainstem, the PPN is located in the mesopontine tegmentum. And the review states it's defined by a cluster of large neurons sitting right adjacent to the decussation of the superior cerebellar peduncle.
Okay, I have to stop you right there, because decussation, that sounds like something out of a medieval medical text.
Yeah, it really does sound incredibly dense, but a decussation is simply a biological intersection.
Oh.
Right, it's just a point where nerve fibers cross from one side of the brain to the other. In this case, the superior cerebellar peduncle is this massive highway of fibers connecting your cerebellum to the rest of your brain.
And the cerebellum coordinates movement, right?
Exactly.
Mm-hmm.
So by sitting right next to this crossing point, the PPN is positioned at like one of the busiest, most critical traffic intersections in the entire human nervous system.
Wow. So it has a front row seat to almost all motor and sensory information passing through?
Yeah, exactly.
Well, that positioning makes perfect sense if its job is to filter all that incoming traffic. But what really stood out in the sources is what the PPN is actually made of.
The chemical complexity, yeah.
Right. The review emphasizes that it's completely defined by the rule of threes, because historically, researchers thought this area was just a cluster of cholinergic neurons.
Right, and those are cells that release acetylcholine, which is a chemical messenger we usually associate with, alertness and attention.
Yeah, but the newer mapping completely redefines that picture, right?
It does. The PPN isn't a monolith anymore. It has three distinct types of neurons, perfectly intermixed in this tiny space.
Okay.
So you have those cholinergic cells, but you also have glutamatergic cells, which use glutamate to act as the excitatory gas pedal for neural circuits.
Got it. The gas pedal.
And then you have GABAergic cells, and those use GABA to act as the inhibitory brake pedal.
So you basically got the chemical gas pedal, the brake pedal, and the steering wheel of acetylcholine all mashed into one structure the size of a pea.
Yeah, which is highly unusual.
Really?
Yeah, because often in the brain you'll find a nucleus dedicated mostly to one primary neurotransmitter to do one specific job.
Right.
But the fact that the PPN has this three-part chemical diversity, that's what allows it to be such an extreme multitasker.
Makes sense. So to really understand how it manages your life, we have to look at its primary job first, which is controlling the state of human consciousness.
Yes. The difference between you listening to this wide awake versus you being completely unconscious in deep sleep.
Right. And the researchers found 3 functional cell types related to this, again, with the rule of threes.
Exactly.
So first, you have wake-on cells that fire only when you are awake. Second, you have REM-on cells that fire exclusively when you are in rapid eye movement sleep-like, when you're actively dreaming.
And the third category is perhaps the most interesting, the wake REM-on cells.
Yeah, I wanted to ask about those.
So these neurons fire intensely during both waking hours and during REM sleep, but they go completely quiet during deep, dreamless, slow-wave sleep.
See, I was trying to figure out why a single cell would fire when I'm awake and also when I'm asleep. But REM sleep is actually a highly active state for the brain, right?
Oh, absolutely. Dreaming is essentially a form of conscious experience. It's just disconnected from the physical world.
Wow, yeah.
The brain activity during REM sleep looks remarkably similar to the brain activity of someone who is awake and, say, solving a math problem.
That is wild.
It is. And the PPN is driving the high-frequency brain waves for both of those active states. And the physical mechanism of how these cells fire is just fascinating.
Yeah, the sources mention that all of these PPN neurons, regardless of whether they use glutamate or GABA, possess intrinsic gamma band activity,
Right.
meaning they naturally fire electrical impulses at a frequency of 40 to 60 hertz. But I want to break down the physics of that for a second.
Okay.
When the review says they have intrinsic activity, does that mean the PPN is basically generating a constant vibrating electrical hum all on its own?
It is a literal measurable hum on an EG. And the way a cell physically generates that hum comes down to voltage-dependent calcium channels on the dendrites.
The dendrites being the branching structures of the neurons.
Exactly. Specifically, they use high-threshold N-type and PQ-type calcium channels.
Okay, alphabet soup again.
Yeah, sorry. But basically, when these channels open, positively charged calcium ions rush into the cell. That changes its electrical charge and triggers a firing impulse.
Right.
Then they pump the calcium out, the charge resets, and the cycle repeats 40 to 60 times a second.
Which is incredibly fast. And the review notes the brain modulates these specific channels using different intracellular pathways to change your state of consciousness.
Yes, exactly.
So for waking up, it uses a pathway called CAM-KI, but for REM sleep, it uses the CAM-PKA pathway.
Right.
I'm gonna need you to save me from this alphabet soup too.
Yeah.
What are CAM-KI and CAM-PKA actually doing inside the cell?
Sure. Think of them as different internal messenger relay systems. The brain sends a signal saying, it's time to wake up. That signal hits the outside of the cell, which triggers the CAM-KI messenger system inside.
Got it.
So CAM-KI runs over and physically unlocks the PQ-type calcium channels. And that starts the gamma hum that keeps you awake.
Oh, I see.
But on the other hand, if the brain says it's time to dream, it triggers the CAM-KI messenger system. And that unlocks the N-type calcium channels instead.
So the brain is just swapping out which chemical key it uses to start the ignition, but the engine always runs at that 40 to 60 hertz gamma hum.
Precisely.
That makes the chemistry a lot more complicated than a simple on-off switch. But for that hum to actually dictate whether you are conscious or unconscious, a few isolated cells humming isn't enough, is it?
No, not at all. They have to work together. They need structural coherence.
Right.
Which brings us to another major finding in the review, the role of gap junctions.
Yeah, the electrical couplings.
Yes. These are physical tube-like connections between the neurons that allow them to share electrical currents and chemical signals directly. They bypass the normal synaptic gaps entirely.
You know, the analogy that came to mind when reading about these couplings was a rowing team.
Oh, that's a good one.
Yeah, because you can have 8 incredibly strong rowers in a boat, but if they are all rowing at different rhythms, the boat just spins in circles.
Exactly.
So these gap junctions are essentially synchronizing the rowers. They are physically connecting the neurons, so they fire their 40 hertz hum in perfect unified lockstep.
That synchronization is everything. And when we connect this to clinical pharmacology, it explains the mechanism behind some of the most powerful drugs we use.
Oh, right. The sources mention modafinil.
Yeah, modafinil. It's a potent stimulant frequently prescribed for narcolepsy. Modafinil actively increases this electrical coupling in the PPN.
So it basically forces the rowing team into perfect synchronization.
Exactly. It decreases electrical resistance across the network, disinhibits the entire circuit, and keeps the patient wide awake and alert.
Wow. And if you want the opposite effect, you just sever the connections.
Yeah.
The sources point out that fast-acting general anesthetics like propofol or halothane actively block these gap junctions.
Right. They take the oars out of the rower's hands.
So the cells might still want to hum, but the coherence is lost, the network shuts down, and you lose consciousness entirely.
It's a profound realization for anyone listening. I mean, if you've ever woken up from surgery feeling that heavy, terrifying grogginess where you can barely string a thought together,
Yeah, the worst feeling.
or conversely, if you felt that hyper alert, jittery focus after taking a strong stimulant,
Yeah.
you are directly experiencing the chemical manipulation of your tiny PPN's gamma rhythm. Your brain's executive assistant has either been handed a massive megaphone or forcibly locked in a soundproof closet.
That is such a vivid way to picture it. Okay, so the PPN wakes the brain up and maintains our consciousness. But biologically, waking up is only step 1, because evolutionarily, we wake up so we can move, to go find food or run away from a predator, which leads to the massive neuroscientific turf war highlighted in these papers.
Oh yeah, the MLR controversy.
Right. If the PPN is controlling our wakefulness, is it also directly controlling our legs?
And this goes back to a famous set of experiments in the 1960s by scientists named Schick and Orlovsky.
Okay.
They found that if they applied electrical stimulation to a specific region in the brainstem of decorticate cats, meaning cats that had their higher brain functions removed, the cats would spontaneously stand up and walk fluidly on a treadmill. And they named this go switch for walking the mesencephalic locomotor region, or the MLR.
And because the PPN is sitting right there in that exact same neighborhood of the brainstem, everyone assumed for decades that the PPN was the MLR.
Yeah, I mean, it seemed like the most logical conclusion at the time.
Right.
But as tracing technology improved, the anatomy started to contradict that theory.
How so?
Remember those cholinergic neurons in the PPN? When researchers actually mapped out where their fibers went, they found that the vast majority project upward into the thalamus and the basal forebrain.
Okay.
They don't project downward into the spinal cord, which is biologically required if you are going to be the central command sending walking signals to the legs.
Okay, and here's where it gets really interesting. The sources dive into a recent study by Sherman and colleagues that flips the entire board over.
Yes, the Sherman study is fascinating.
Because they went looking for the neurons that actually do project directly down to the spinal cord. And they found them, but they weren't the PPN cholinergic neurons.
No, they weren't.
They were a separate population of glutamatergic neurons located just 1 to 2 millimeters medial to the PPN.
A completely different set of cells sitting right next door.
Right. And to prove what these specific cells did, Sherman's team injected a glutamate analog directly into this neighboring medial area in awake, freely moving rats.
And the result was bizarre.
Seriously bizarre. The rats started walking like robotic automatons. They just marched forward completely devoid of normal exploratory behavior.
Yeah.
They walked until they hit the wall of their enclosure, and because their brain was artificially screaming, "Move forward," they literally kept trying to step vertically up the wall.
It's a striking demonstration of how deeply hardwired these lower brain circuits are.
Yeah.
When you hit the raw go switch, the animal loses all context. It just executes the motor program.
Wow.
So the Sherman data provides very strong evidence that the true MLR, the actual initiator of stepping, is that specific population of glutamatergic neurons sitting adjacent to the PPN.
Okay, I have to push back on this then.
Go for it.
If the true MLR is next door, making the physical legs move, what is the PPN actually doing for our movement?
That is the $1 million question.
Right. Why has it been implicated in walking for 50 years? Is it just guilt by association because they share an anatomical zip code?
No, it's much more than just proximity. And this gets to the heart of how we define locomotion. The consensus review is very careful to clarify that the PPN is not a crude on-off switch for stepping. Instead, it acts as a rhythmogenic center.
A rhythmogenic center.
Right. It sends descending projections to other areas, like the reticulospinal systems in the lower brainstem, to modulate your muscle tone, your posture, and the cadence of your gait.
Oh.
And it does all this in parallel with managing your arousal.
That makes a lot of sense, actually, because you don't walk in a vacuum. You need the conscious spatial awareness to walk safely without marching into a wall like that rat.
Precisely the point. The PPN is an integral part of the reticular activating system.
Okay.
It coordinates the internal state of being awake with the physical act of moving through space. It bridges consciousness and motor control.
So it's providing the rhythm.
Exactly. The PPN provides that synchronized 40 hertz gamma hum to organize the entire behavior. It ensures that your posture is stable enough to support the stepping signals coming from the MLR next door.
Wow. Well, if the PPN is the central coordinator for the rhythms of walking and posture, that brings us to a really critical clinical application.
Yes, it does.
What happens when those rhythms are fundamentally broken, like we see in advanced Parkinson's disease? The sources discuss using deep brain stimulation, or DBS, to essentially hack the PPN.
Right, because Parkinson's disease can lead to a profoundly debilitating symptom known as freezing of gait.
Which sounds awful.
It is. Patients describe it as feeling like their feet have been suddenly superglued to the flare. They have the conscious desire to walk, but the motor command just won't execute.
And standard medications don't work for that.
Often they fail to alleviate this specific symptom, yeah. So neurosurgeons began implanting DBS electrodes deep into the brainstem, targeting the PPN area, to try and electrically unfreeze these patients' neural circuits.
Well, wait, let's look at the spatial physics of this procedure based on what we just discussed. We know the true MLR is only 1 to 2 millimeters away from the PPN. And the review states that a standard DBS electrode is about 1 millimeter wide, and the electrical field it generates extends about 1 millimeter on either side.
Right.
So aren't we basically driving a bulldozer through a garden here? If the target is that microscopic, aren't surgeons inevitably stimulating the PPN, the true MLR, and all three cell types simultaneously?
You've identified the exact reason why the clinical results of PPN DBS have been incredibly inconsistent and sometimes highly perplexing.
I can imagine.
It is a blunt instrument applied to a densely packed microscopic neighborhood. Because the electrical field is hitting so many different networks at once, patients experience wildly diverse side effects.
Like what?
Well, a perfect example highlighted in the text. Some early surgical teams were stimulating this region using low frequencies, around 10 to 30 hertz.
Okay.
And in several cases, simply turning the stimulator on instantly induced sleep in the patient, right there in the operating room.
Oh my God. You were trying to cure a patient's inability to walk, and instead you put them to sleep. That has to be terrifying for a surgeon.
Very much so.
But it makes complete sense based on the math. The PPN's natural rhythm for wakefulness is that 40 to 60 hertz gamma hum.
Exactly.
If you artificially feed the circuit a slow 15 hertz rhythm, the brain interprets that as a signal to power down into slow wave sleep.
It's a brilliant deduction. The physiology dictates the outcome.
Wow.
And when neurosurgeons realize this and adjust to the stimulation to match the natural gamma frequency, so running the electrodes at 40 to 60 hertz, they began seeing much better, more reliable result.
For improving posture and reducing the freezing.
Yes. The consensus review strongly suggests that the future of this therapy relies on adjusting the surgical coordinates slightly medially and ventrally.
To get closer to the true MLR.
Exactly. Get closer to the glutamatergic MLR neurons to initiate movement while carefully matching the PPN's high-frequency rhythm so you don't accidentally disrupt the patient's state of consciousness.
It really emphasizes that you can't just shock the brain. You have to speak its exact mathematical language at the correct frequency, or the entire system falls apart.
Absolutely right.
So what does this all mean? We've covered a structure that regulates the chemical keys for our sleep and generates the electrical hum of our waking consciousness.
Right.
We've seen how it acts as the rhythmic coordinator for our posture and gait. But the review points out that there is one overarching job that ties all of these functions together.
Yes.
The PPN is fundamentally the ultimate sensory filter for the brain.
Yes, it is. And the formal clinical term for this filtering mechanism is sensomotor gating.
Sensorimotor gating.
Researchers measure the health of this filter using a neurological phenomenon called prepulse inhibition, or PPI. And the PPN is the primary brainstem structure responsible for mediating this.
Okay, let's look at prepulse inhibition, because this is where the analogy of the ruthless executive secretary really crystallizes.
Let's hear it.
So let's imagine the thalamus, which sits just above the brainstem, is the CEO of your brain. The CEO needs to focus on high-level cognitive tasks. The PPN is the secretary sitting at a desk in the antechamber outside the CEO's office.
Right.
Every single sensory input, a visual flash, a shift in temperature, a sound, comes to the secretary first. The PPN looks at the memo and decides, is this threatening enough to interrupt the boss, or can we just file this in the trash?
That captures the essence of the filter perfectly.
Really?
Yeah. Prepulse inhibition is how we physically observe the secretary working. So imagine you are in a quiet room and a door violently slams behind you.
Okay.
Your natural response is a massive full-body startle reflex. Your heart rate spikes, your muscles tense.
Oh, sure. I jump out of my skin.
Right.
Yeah.
But if you hear a floorboard creak just a few milliseconds before the door slams, a healthy brain will actually suppress that massive startle reflex.
Because the secretary caught the warning sign.
Exactly.
The floorboard creak is the prepulse. The PPN processes that tiny sound, realizes a larger event is likely coming, and sends a memo up the chain saying, I've detected a shift in the environment, brace for impact, but don't freak out.
You've nailed it.
It actively gates.
Or screens out the full intensity of the external sensory noise so that the higher cortical regions aren't overwhelmed by every single stimulus.
Wow.
It maintains your cognitive stability.
Wow.
Without the PPN functioning properly, the filter collapses and you would jump out of your skin at a dropping pin. Your brain would be crushed under the weight of its own sensory inputs.
Which brings us to a fascinating physical reality noted in the review about how this filter actually operates in our daily lives.
The posture connection.
Yes, because I was thinking about when I am most alert versus when I'm relaxed. If I lie down flat on the floor, my brain naturally starts prepping for sleep. We don't sleep standing up.
Generally not, no.
The sources suggest this gating mechanism is physically linked to our posture.
It is. The connection between posture and arousal is hardwired directly into the PPN.
So how does that work?
Well, humans naturally sleep in a supine flat position, but we are most awake, most cognitively engaged, and required to process the highest volume of sensory information when we are standing upright, moving through the world.
Right.
So the PPN neurologically links the physical act of verticalization, which is the muscle tone required to stand up, with a massive instantaneous increase in sensory processing and arousal.
So the simple mechanical act of standing upright physically turns the secretary's alert level from a 2 to a 10.
The posture dictates the arousal level, and the arousal level dictates how aggressively the filter processes information.
That is mind-blowing.
They are completely inextricable inside the circuitry of the PPN. You cannot change one without affecting the other.
That really brings this entire exploration full circle.
It really does.
I mean, we started by looking at this tiny pea-sized hub defined by a complex mix of 3 different cell types.
Yep, the rule of threes.
Right. We broke down how the rush of calcium ions generates the 40 to 60 hertz gamma waves that form the literal electrical backdrop of our conscious waking thoughts and our deepest dreams.
It's a gamma hum.
Exactly. And then we waded into the decades-long MLR turf war, realizing the PPN isn't the crude go switch for walking, but the sophisticated rhythmic coordinator ensuring we have the posture to move safely.
Correct.
And finally, we see its ultimate role, the brain's executive secretary, filtering the chaotic noise of the physical world, deeply tied to our physical stance.
It is a masterclass in biological efficiency. But, synthesizing all of this raises a crucial, somewhat provocative question that I think listeners should ponder long after this deep dive in.
Okay, lay it on us.
Well, if this microscopic region, the PPN, intrinsically links our upright standing physical posture with our brain's alertness and its ability to filter out sensory noise, what is our modern environment doing to us?
Oh, wow. I see where you're going with this, and it's a little terrifying.
Think about the average daily routine today. Sitting in ergonomic chairs for 8 to 10 hours, slumped forward over a keyboard, staring at a fixed point on a screen.
Yeah, that's pretty much everyone.
Right. So verticalization is required to optimize our arousal and sensory gating. Could our intensely sedentary lifestyles be fundamentally rewiring how our PPN filters reality?
Because we aren't maintaining the upright posture the PPN evolved to manage.
Exactly. Are we chemically and electrically confusing our brain's secretary just by the way we sit? Are we artificially dampening our own cognitive filters?
That is a deeply unsettling yet incredibly profound thought to leave on. Are we inadvertently breaking our own internal executive assistant just by chronically slouching at our desks?
It's definitely something to think about.
It definitely makes you want to stand up and recalibrate that gamma hum. Well, we want to thank you so much for joining us on this deep dive into the brainstem. We hope it gave you a deeper understanding of the microscopic machinery that is constantly running your life behind the scenes. Keep asking questions, maybe pay a little more attention to your posture today, and as always, stay curious.